So in this recording, what we want to focus on is DNA replication. So we had talked about the structure of DNA and how it lends itself to its functions. And one of those functions is its method to replicate itself. And this all comes back to the idea that DNA, in its most standard form, is a double-stranded molecule with complementary base pairing. So Watson-Crick base pairing is going to be the key to understanding how DNA replicates. And because I can take a parent molecule and pull those two strands apart, now immediately I have two templates to create a complementary strand. So this model of replication, which to our understanding is the most accurate one, is known as the semi-conservative model. One could postulate different models, the conservative model, where the parent molecule makes a daughter molecule, but nothing physical from the original molecule is found in the daughter molecule. In the semi-conservative model, you'll notice that one of the strands actually now is one of the strands in the daughter molecule. The dispersive model is another alternative where the parent molecule is then split up not strands specifically among the daughter molecules. But the semi-conservative model is the one that we're going to work with. Now, DNA replication begins at what are called origins of replication. Okay, so this is where we talk about a replication bubble opening. So eukaryotic chromosomes, and a chromosome is just a big piece of DNA, may have hundreds or even thousands of origins of replications. Note that a eukaryotic genome, and genome is the collection of all the DNA in an organism. So for example, humans have a genome that's about 3.1, 3.2 billion base pairs, whereas E. coli has a genome of about 5 million base pairs. So the difference between prokaryotic life and eukaryotic life is several orders of magnitude with regard to genome size, though genome size varies considerably among the eukaryotes and prokaryotes. So what we're going to see is replication proceeding in both directions, because replication is going to be only done 5' to 3'. And we'll get to that in just a minute. But you can see here in a circular chromosome of a prokaryote, you have an origin of replication. How does the machinery that is going to engage the sequence that denotes the origin of replication, how does it recognize it? Through the major and minor group. So interacting with that sequence through the major and minor group says, oh, this is where I'm supposed to start to pull the strand apart and start to build new molecules of DNA. And that simply then runs in both directions until we have two new daughter molecules. Well, if you have a linear chromosome, like we see in eukaryotic cells, you have several bubbles of replication starting, several origins, until they all meet to get our two daughter molecules. So what we need to do now is break down the players in this event and start to look at the different proteins that are involved and start to learn the terminology. So at the end of each replication bubble is what's called a replication fork. And so this is where a lot of machinery is going to be found. And so we're going to have helicases. These are machines, they're little proteins that untwist the double helix. We have single-stranded binding proteins that are going to stabilize single-stranded DNA so it can be used as a template. Because again, remember, Watson-Crick base pairing is not specific to an alternative strand. If a DNA molecule twists back on itself, if an A finds a T, it's going to bind. All right, so we need to keep single-stranded molecules, if we're going to use them as a template, kind of stretched out nice and neat so it can't fold back up on itself. Now we're also going to find a cool molecule called a topoisomerase. And that topoisomerase is further upstream of the replication fork, and it deals with the twisting of the DNA as you unwind it. And so what you do is you create tension in the DNA molecule that you need to relieve, and topoisomerase does that. So a great video to watch to look at the real-time kind of replication of eukaryotic DNA is this one. If you click on that link, it'll give you some great views of computer models of DNA replication. Let's kind of drill down and start to look at some of this machinery. So we can see here the topoisomerase, single-stranded binding protein, the helicase, and a new protein that we haven't talked about yet called the primase. We'll get to that in just a minute. But basically, DNA polymerase 3, which is the eukaryotic DNA polymerase that's going to replicate DNA can only add a nucleotide onto an existing nucleotide. So you have to prime the strand, and primase is what's going to do that. Note that in all of these pictures, and I want you to get into that habit of noting, is the orientation of each molecule, 5' to 3', 3' to 5'. We're always going to build DNA 5' to 3'. We can only add on to the 3' end. So the enzyme that builds DNA is called DNA polymerase. So what does it do? It polymerizes DNA. So in bacteria or prokaryotes, this happens much more quickly than it does in eukaryotes. Human DNA polymerase runs about 50 nucleotides per second, whereas it's 10 times as fast in a prokaryotic cell. Also, prokaryotic cells tend to be more error-prone, but that also is to their advantage, considering the way they reproduce, and we'll talk about that more as we talk about evolution. So a nucleotide is added to the growing DNA strand. Now, this is where you have to be specific, right? Are we talking about DNA or are we talking about RNA? So we're talking about DNA now. So we're going to add dATPs, dTTPs, dGTPs, gCTPs. Now, we haven't talked about ATP as an energy molecule yet, but we will. But the ATP is adenine triphosphate. So there's 3 phosphates on there. And again, we're going to note the difference between RNA and DNA based on the sugar that's there. So the best way to do is to look at this. And we're actually going to break off 2 phosphates from the trinucleotide phosphate, from the nucleotide triphosphate, and use that as energy. Right? That's going to be energy to catalyze the reaction, and that's going to enable us to build our strand. Notice that we always add then to that 3' hydroxyl group to make that phosphodiester bond. All right? Now, as I mentioned before, we have a primer necessary for DNA polymerase. So it can only add nucleotides to a 3' end. So if you've got nothing there, you can't build. So this is going to cause an interesting problem later, but we'll talk about that when it comes to the ends of the DNA, linear DNA molecules that we're trying to replicate. But primase is going to come in, and it's going to put a little RNA chain down, and that primer is just enough, 5 to 10 nucleotides, for DNA polymerase 3 to start adding on new nucleotides. All right? Now, remember, DNA is anti-parallel, so we're going to head off in both directions. But at a particular fork, replication fork, what you're going to see is a leading and a lagging strand based on this. So you can see there here, this is a great picture, because what you can see is if we start at the origin of replication, if we head to the left, the top strand is the leading strand. You're building a new DNA molecule 5' to 3'. But the opposite strand, the complementary strand of the original molecule, you can't build it in the same direction the replication fork is moving away from the origin of replication. So you have what are called Okazaki fragments. And these Okazaki fragments then are going to have to all be individually primed, and then later the primers are going to have to be removed. It's all going to have to be kind of stapled together by an enzyme we call ligase. So all that extra work results in what's called the lagging strand. And you would have seen that in the video had you looked at that. Now, again, if we look at the machinery here, we definitely see here's the replication fork. There's our RNA primer, and we have our DNA polymerase 3 moving away from it, adding new DNA to complementary sequence. This is basically how it works. There's not a whole lot more going on there. Again, the lagging strand has called Okazaki fragments, so the series of segments that are ultimately going to be joined together by DNA ligase. Right? So in this case, you have Okazaki fragment 1, Okazaki fragment 2. And again, if you can go back to that video, you can see definitively how that lagging strand looks very, very different as it's kind of looped around to be replicated, as opposed to the leading strand, which is kind of one continuous addition of DNA. Now, the pieces, the players here, are important to know, and this is a great chart to kind of summarize them. The helicase, single-stranded binding proteins, topoisomerase, primase, DNA polymerase 3, polymerase 1, and DNA ligase. So let's look at all those kind of in one big picture. And this is a great picture to be able to kind of reproduce if you truly understand all the players that come with DNA replication. So we can see down here, DNA polymerase 1, which is something we haven't mentioned yet, it's actually going to come in and it's going to chew away at the primers, removing the RNA and laying down DNA in that little 5 to 10 nucleotide region. Now, that's cool, but what you have then is separate pieces of DNA that are complementary to their template, but aren't one big molecule. And so finally, in comes DNA ligase, which is going to kind of staple together the backbone of those two adjacent oligonucleotides, and now you have one single molecule of DNA complementary to the original template. We're going to look at ligation and the polymerases in a little bit more detail as we go further on.